Although peripheral blood lymphocytes and monocytes do enter the CNS from the circulation and patrol its tissues, the primary immune effector cells, permanently residing within the central nervous system, are the microglia. They are believed to derive from bone marrow precursors of the macrophage monocyte lineage and to have migrated into the CNS during the perinatal period, around the time of birth. Microglia are activated by brain injury, infection, or neuronal degeneration. Their activation response is multidimensional, with several changes occurring simultaneously. They proliferate and migrate into damaged tissue. They increase or newly express surface markers including CD45, also called leukocyte common antigen, as well as MHC class I and class II molecules, and immunoglobulin Fc receptors. The expression of MHC class I and class II molecules is particularly significant because it means activated microglia can present antigens directly to T lymphocytes, effectively linking the innate immune response within the CNS to the adaptive immune system. The Fc receptors allow them to recognize and clear targets coded with antibodies, further extending their immune capabilities. Alongside these surface changes, activated microglia secrete cytokines, reactive oxygen intermediates, and proteinases. This entire response is designed to remove dead tissue and eliminate invading pathogens. However, the same destructive tools, especially reactive oxygen species and proteinases, can damage surrounding healthy tissue in the process. This dual nature of microglial activation explains why it is implicated not only as a protective mechanism, but also as a contributor to tissue damage and disease progression in inflammatory and degenerative CNS conditions. In summary, microglia are the resident innate immune cells, macrophages, of the central nervous system, CNS, constituting about 5 to 15% of all brain cells and serve as the CNS's primary immune effectors responsible for homeostasis, immune surveillance, synaptic pruning, and responses to injury or disease. They constantly survey the neural environment, rapidly sense damage signals, and, upon activation, can become either neuroprotective, clearing debris, supporting repair, promoting remyelination, or neurotoxic, exacerbating neuroinflammation and tissue injury, depending on context and activation state. Microglia derive from embryonic yolk sac precursors, infiltrate the CNS early in development, and become self-renewing, long-lived cells. In normal CNS, microglia regulate synaptic pruning, neuronal proliferation and survival, and orchestrate programmed cell death without causing inflammation, phagoptosis. Microglial dysfunction activation contributes to diverse CNS pathologies, including multiple sclerosis, Alzheimer's disease, amyotrophic lateral sclerosis, perinatal white matter injury, demyelinating diseases, and chronic pain syndromes.